right. Welcome everyone to the RNA Collaborative seminar. This time it's organized by the Bay Area RNA Club. And we're very happy to welcome you all to this seminar. And we have two speakers today, Gloria Brar from UC Berkeley and Rhiju from University of Stanford, that we're really excited to hear their latest updates on their research. So I don't want to stretch the introduction too much. There's one little thing that I do have to mention in terms of administration. So if there are any questions, and we're obviously very excited to hear your questions, there's a Q&A button on the bottom that you can all use. So please type in your questions down there, and I'll be able to read them out after or in between the presentations. So with that, I want to quickly introduce our first speaker of the day, Gloria Brar. So it's a really great pleasure for me to introduce Gloria today. Generally speaking, Gloria's lab studies the gene regulation circuitry that underlies meiotic differentiation. But more specifically, she had elucidated many different forms of meiotic regulation. For example, regulation through mRNA translation, timed protein degradation, or through peculiar short proteins, and also through alternative RNA isoform expression. So we're very excited to hear more about that. Let me give you a little bit of Gloria's background to you as well. So Gloria did her PhD with Angelica Amon at MIT, and later joined Jonathan Weissman at UCSF for her postdoc. After that, she had started her own group at UC Berkeley in 2014, where she continues to conduct very exciting research up until now. And yeah, with that, today she's going to talk about the genomic expression cassettes driving unexpected RNAs and their off-target effects. And yeah, with that, I'd like to hand over to Gloria. I'm really looking forward to her presentation.
Thank you so much, Stefan. I really appreciate it. Can everyone see my screen okay?
Looks good.
Looks good. Okay, thank you so much. All right, so let me just hide these controls if I can. There we go. That'll be better. Okay, so thank you so much, Stefan, for the invitation. I'm really excited to be here. This is a really great super group with a lot of people who care about RNA from kind of diverse, dispersed backgrounds. And what I'm going to tell you about is a bit of a strange story for my lab. This was not a project we were trying to work on. This was kind of a weird observation that just came from a really great lab member, Emily Powers, who saw something weird was going on in her data and decided to follow it. And I think what she found has really wide-ranging implications for how we think about genome organization and genome engineering strategies. Oops. So what I'll be talking about today is an issue that arises from the use of expression cassettes. And so I just want to briefly tell you all something you probably know very well, which is that we really think of regulatory regions of the genome and coding regions of the genome to be fairly modular. And so pretty early on, it was found that you can kind of put these together in different combinations and make these kind of cassettes that can be useful for a number of purposes. And so I'm just showing one example of a really simple case here where you have some sort of minimal promoter, NFI' UTR, and then you can put some coding sequence of choice in here. And as long as you put kind of a 3' UTR and terminator, this should be able to express pretty well. And then you can do all sorts of things with this type of construct. You can use this to either increase or change expression of whatever your gene of interest is, which you would put right here. You can make more complex constructs that include epitope or fluorescent tags or mutations. You can make reporter constructs. And you can also use this general strategy to delete genes of interest. And in all of these cases, these constructs might be outside of the chromosome or they might be genomically inserted. And I think generally it's thought that genomic insertion is kind of the more stable way to do things in a lot of cases, as long as it's done really, really precisely. And I should say there's some differences in kind of how we do this in different organisms, but in general, this type of strategy is used pretty broadly. Now, where our story started was with Emily Powers, just an absolutely fantastic scientist who was doing something kind of mundane. She was using an expression cassette approach to replace a gene of interest. So we work primarily in budding yeast, and the way that this is typically done in our organism is using a set of plasmids that have been used by, hundreds to thousands of labs, many thousands of times to make kind of very targeted changes in the yeast genome. And so the way this works is we have a plasmid that has kanamycin resistance gene, for example, under yeast control. So here it's the TEF promoter and terminator. And so what Emily did was to amplify up this cassette and use primers that have homology to the region flanking what she wants to replace. So basically she does a PCR, gets this piece of DNA out, and then transforms this into cells. And through cellular repair pathways, now this gene of interest will be replaced in a small subset of cells by this kanamycin resistance cassette. And then we can, of course, select for these cells. And so this is a very precise way to delete a gene. And typically we would just replace the entire coding region, and that's what Emily did. And the gene that she deleted was one that might be of interest to this audience. This was DBP1. This is an RNA helicase, a dead box helicase. It's the paralog of the more famous dead one, which is DDX3 in humans. And this is a RNA helicase that's really important for translation. Now, as Stefan mentioned, my lab does a lot of work looking at meiosis. And one thing that Emily had noticed is in meiosis, DBP1 is actually fairly highly expressed and DED1 expression goes down. And this is in contrast to almost every mitotic condition where DBP1 is not expressed at all. And so what Emily thought is that the kind of partial replacement of the RNA helicase pool of DED1 with DBP1 and meiosis might be controlling translation in some special way in this context. And so that was the reason she wanted to delete this gene. So she did a number of assays. What I'm showing you here is some of her genomic data. She did RNA-seq and ribosome profiling. I'm just showing you the data over this region that contains the DBP1 gene. We have just kind of a quality control check here showing that indeed we're not seeing DBP1 expression at the mRNA or translation level here compared to wild type cells. But Emily, who is an incredibly careful scientist, noticed something else, and that's that this neighboring gene seemed to have something weird going on specifically in these cassette replacement strains. And I should say this cassette that Emily inserted at the DBP1 locus did not disrupt any known regulatory region for the neighboring gene. And so we didn't expect anything to be happening there. But if I zoom in, hopefully you can see there's a kind of subtle change there. There is an apparent 5' extension to the transcript for this neighboring gene, which is divergent in its orientation relative to DBP1. And when we look at the ribosome profiling data, we see translation in these little regions that represent UORFs, these initiated AUGs that are in this 5' extended region of the transcript. And this seems to be associated with low translation of the ORF for the neighboring gene. And so this was a little strange, but what Emily did was to confirm that what it looked like was happening here was really happening. And so the way she did this was initially a 5' race, just looking at the 5' ends of the transcripts for this neighboring gene, MRP51. And what she found was that indeed, when this DBP1 ORF was replaced with this expression cassette, now she saw a change in the 5' end of MRP51. And this is upstream of these UORFs. And so the model for what seemed to be going on was that in wild type cells, this neighboring gene, MRP51, makes a normal transcript and is translating MRP51 protein. But in these cassette replacement strains, it looked like a different 5' UTR is here now due to an upstream transcription start site this new transcript includes UORFs, and this dampens the translation of the downstream ORF. And this was something that was very interesting to us because this looked like a kind of natural regulation that we'd spent a lot of time studying. And this regulation can be summarized here. This was first defined by Elchin Unal's lab, and then our lab has subsequently done a lot to look at this. And the idea here is that we've found that hundreds of genes in the yeast genome, and especially in meiosis, are regulated through this strange strategy. Again, the difference here is this is natural, whereas what we're seeing in this other case seems to be unnatural. But the way the natural regulation works is that these genes have two different transcription start sites. And in a temporally regulated way, there can be switching between use of these two sites. In one case, if the canonical site is used, a normal transcript is made that can make protein. But if the upstream site is used, instead of making a canonical transcript, cells make a 5' extended transcript, which we term a LUTI, or long undecoded transcript isoform. And this is very poorly translated due to UORF translation. And what we see empirically is that the switching between these two isoforms is controlled by regulated transcription factor expression that drives the two isoforms, and that when more of the canonical transcript is made, we see protein being made. When more of the LUTI transcript is made, we see protein production being halted. And this is due to this UORF translation, but also because production of these LUTIs silences this downstream transcription start site in cis, and so stops the cell's ability to make a translatable transcript. And so this is actually a way to make a transcript that shuts down translation. And that's what we thought was maybe happening when we inserted this cassette next to MRP51, although we didn't really understand why. And so Emily did some checks to see if the features that we had defined for this natural regulation were being artificially induced in this cassette replacement case. And one thing she did was to look at her global ribosome profiling data. She saw that indeed DBP1 was, of course, very reduced in its translation, but MRP51, this neighboring gene, was actually greatly reduced as well, one of the most downregulated genes. And this was despite the fact that the total mRNA levels for MRP51 were up. This is actually a hallmark that we see of this LUTI regulation. There's often a lot of mRNA. It's just the wrong kind of mRNA. It contains the coding region, but it can't make protein. And so this then results in a really low TE, or translation efficiency measurement. This is a measurement we use with ribosome profiling data a lot. The idea is this is the ribosome footprints normalized to the amount of mRNA. So this is kind of footprints over mRNA. And when we look at this globally, what we see is that in these cells with the DBP1 replacement, MRP51 is one of the most translationally downregulated genes. And this does affect protein production, although interestingly, maybe not as much as we might have expected. There is a significant and reproducible decrease in MRP51 protein relative to wild type cells. And so we were getting a little nervous here. This was clearly an off-target effect, and we didn't know if it might matter. And I should say at this point that MRP51 is a mitochondrial ribosomal protein. And so we wouldn't necessarily expect it to have any effect on translation, but we wanted to revisit the mutant phenotypes we had seen to determine whether this might be causing some issues. Some of the phenotypes I was pretty confident about. For example, DBP1 is, again, supposed to be a translation factor, and RNA helicase important for translation. And when we looked in meiotic cells deleted for DBP1 in this way, we saw a decrease in polysomes. And this seemed to represent a decrease in bulk translation as assessed by S35 amino acid incorporation experiments. Also, these cells were bad at completing meiosis as assessed by sporulation efficiency measurements. So it matched our overall hypothesis, which is that DBP1 is affecting translation and then affecting meiosis. But Emily had seen some other mutant phenotypes that didn't quite make sense and were now making her very nervous. And these were based on mitotic growth of these strains. So when she grew these strains mitotically in media containing glycerol, or in a media containing dextrose at late stages, that is in the post-dioxic shift, which is when these cells start to require respiration. And I should say in glycerol media, these cells require respiration. What she saw was that these DBP1 replaced strains had poor growth. And this is clearly something that could be mitochondrially associated. Moreover, DPP1 shouldn't be involved in this at all because it's actually not expressed in mitosis in most conditions. And so Emily went forward to try to determine if any of these mutant phenotypes were dependent on DPP1 or if they were all due to the side effect. And what she found, to our surprise, was that indeed, every mutant phenotype she could find in these strains wasn't due to the complete removal of DPP1. It was due to the slight decrease in MRP51 protein that came as an off-target effect of her precise engineering approach. So here we're looking at strains where she replaced either DPP1 or MRP51. What you see is the respiratory growth is rescued by the replacement of MRP51 in both these cases, and that's maybe not surprising. But this is also true of sporulation efficiency and actually this translation phenotype that we saw. The poor translation in meiosis was rescued by adding back MRP51, but not by adding back DPP1. So we started to wonder if we had just gotten really unlucky or if this might be going on in other places. And the first thing we did was to look at a published data set that had proposed a specific function for DPP1 based on this type of cassette replacement approach. And this was doing this in a different strain background with a slightly different cassette and a very different lab. But I'm showing you their genomic data here, and hopefully you can see that they actually saw the exact same thing. They saw this 5' extension with UARF translation, but just hadn't really noticed in the initial analysis, which is understandable given what a strange thing this is to see. Moreover, they saw this really dramatic decrease in MRP51 translation, but not mRNA. So this seemed to happen as far as we can tell, every time someone inserts a cassette at the DPP1 locus. So then we tried to test if this was really true. If we put in other cassettes, does this keep happening? And indeed, no matter what the composition of the cassette, different promoters, different genes, different terminators, we always saw this defect in MRP51 caused by this aberrant RNA, which was easiest to assay by just looking at respiratory growth. So then what Emily did was to try to see if she could get this to go away. And so she put little minimal terminators at either end of this cassette, either from DEG1 or SIC1 to act as kind of insulator elements here. And the idea was she thought maybe this would rescue the normal isoform of MRP51. And indeed, she found that it did. So by 5' race, the 5' end went back to normal for MRP51. And actually, everything was rescued in this strain. The protein abundance of this mitochondrial ribosomal protein, as well as the growth in this non-fermentable media requiring respiration. And so then she asked, what is it about the cassette? So what she did was to reverse its orientation. And she saw that when she did this, now actually there was no longer this side effect. So it wasn't something about just having the cassette there, expressing at very high levels in the specific place. It seemed to be something actually about the promoter of the cassette. Again, not specific to the TEF promoter, but just whatever the promoter was. And so what she did now was to just test this by just insulating the promoter with one of these little terminator elements. And again, whether it was forwards or backwards, she saw that this rescued the issue. There was no longer the misregulation of the neighboring gene. And so it looks like the promoter is the problem. So at this point, we wanted to understand what is going on. And we started to think that this was actually maybe obvious because it's been known now for 10, 15 years through a series of beautiful papers that essentially all promoters are bidirectional. So this was first seen in Giardia and then yeast and human through beautiful papers, including this one by the Steinmetz lab, that promoters inherently have bidirectional activity. And so we thought maybe this is what's going on then. These promoters that we're inserting are driving divergent expression that we just didn't know to look for. And so we looked back at data sets that we already had where we had inserted cassettes at different places in the genome. And that included this data set here where we replaced this NMD gene UPF1 with a cassette. And what we saw is that in some cases, we clearly could see evidence of this divergent transcription. So here you can see this divergent transcript only in the cassette replacement strain. But this was actually not true in the majority of cases. So only 5 out of 19 deletions we analyzed had a clear divergent transcript. And so we wondered, why is it such a low number if this is just an inherent property of promoters? And so now we turned again to something that should have been maybe obvious from the literature, which is that at their endogenous loci, their most divergent transcripts actually are dealt with by early termination signals. And so this is a little bit different whether we're looking in yeast or human, but again, a lot of really great papers that came out in kind of a flurry showed that in yeast, there's this alternate transcription termination mechanism, the NNS pathway, which is usually used to terminate short RNAs like snow RNAs, seem to also be very important for terminating these divergent transcripts so they didn't get too far and muck up neighboring gene expression. And then these transcripts, these short transcripts, were targeted for degradation by the exosome together with with its catalytic subunit, rather, RRP6. A similar mechanism seems to be there in humans, although different factors are controlling it. And the idea is that these sequences to cause early termination have evolved to minimize the effects of divergent transcription. That's just an inherent property of promoters. So what we started to wonder then is this happening every time we insert a cassette, and maybe we just can't see the evidence because these transcripts are being targeted when they happen to be inserted somewhere that has early termination sites or NNS recognition sites. And so what we did was to look in the literature at all the data sets we could find where something was replaced with a cassette and RRP6 was removed. So we have 2 examples here from the Chamfreau lab and the Steinmetz lab, mRNA-seq and tiling array data. And what you can see in both these cases is that the gene that's deleted doesn't show evidence of a neighboring divergent transcript unless RRP6 is also deleted, in which case now we do see a robust short divergent transcript. So we wondered then if we could fix this problem by, instead of using a minimal promoter, which is historically what's been done, if we used the whole endogenous region around one of these promoters. So the TEF gene actually, like essentially every gene, is known to have divergent transcription. And this was shown by the Steinmetz lab. By deleting RRP6, you can see this divergent transcript pops up, but the minimal promoter doesn't have this region that should include these early termination sites. And so what Emily did was to clone out just the short minimal promoter for this gene and also this longer region. And then she also just took the normal TEF promoter, which is actually from a slightly different species, and put just another NNS sequence there from a snow RNA. And when she did this, she found now there was no evidence of this side effect that we had seen. So these, either including the normal promoter context or just an NNS sequence, blocks the ability of this divergent transcription to get into the neighboring locus and cause problems. So to kind of summarize then what we found, bidirectional promoter activity is retained when we port minimal promoters into other places. And this is, of course, something we do all the time in all sorts of different organisms. And when you take it out of its native locus, you're taking away these fail-safes that have evolved to limit the problems with divergent transcription. And you can get side effects of changing promoter context in kind of an unpredictable way, depending on where you've inserted that promoter. And this can dramatically affect neighboring gene expression. And this is probably, we think, being pretty widely overlooked. I should say all features of what we see here in yeast are shared by mammals. And so we think this is going on in some situations there as well. And I should say it's really hard to look for this actually, because it can be condition specific. So I should say in the case of DBP1, we didn't see any evidence of this when we looked in rich media because those cells didn't care about MRP51. And it was only under these specific growth conditions where mitochondrial activity was important. So I should say in yeast, we have these new cassettes, which don't cause problems. So if people want them, you're welcome to them. And we would say in other systems, it's probably worth looking for this and thinking about this in the design of strategies. So with that, I have to thank Emily, who did everything and was just incredible. Thank my group. I run a team lab with Elchin Unal and Charlene Chan, a great technician in the lab, as well as Lydia, a former rotation student, contributed a lot to this project as well. And we collaborate a lot, of course, with Elchin, but also Marko Jovanovic. So thanks. I'd be happy to take any questions.
All right. Thank you so much, Gloria, for that super nice presentation. So we have one question already, or we have a few questions popping up in the chat. I'll quickly read them out. One question from Ben Montpetit is 5 out of 19 is not a majority, but what do you expect this could mean for functional genomics data that is abundant in yeast using the deletion collection? Yeah. SGA, chemical genomics, etc.
Yeah. Hi, Ben. Great question. Yeah, so that's a really great point. And I should have said also that we are not the first people by any means to notice that sometimes weird things happen in the neighborhood of genomic insertions. So Martin Kupiak has published a couple papers on this and some others. Actually, I don't know if I can show it. Sorry, I have a deck of slides, but this paper is a favorite. It might be published now. I haven't updated my slide, but this was a bioarchive paper from last year where actually this group did mass spec on the deletion collection. And what they noticed was that genomic neighbor changes in protein levels were about as common as common an explanation for genomic insertion effect as known protein-protein interactions, actually. And so when we try to estimate how common this might be a problem, we think in the neighborhood of 7%. Based on studies like this one and also some of the work from Martin Kupiak. Yeah, it's a little hard to know. The thing that's disturbing is that as far as we can tell, you can't predict when it's going to happen. Just looking at a locus, we can't tell. And so that's what kind of worries me. But it will be the minority of cases for sure.
Okay. So yeah, the second question was along the same lines, basically asking how pervasive do you expect misassigned side effects to be in the literature? If you do want to elaborate a little bit more, though, I think.
Well, yeah, I mean, I think, so I should say there are certain, what we see specifically, this kind of neighboring gene downregulation of translation, which is, I think, something none of us would have necessarily thought to look for because transcript abundance again stays the same or even goes up. This you can predict to some extent because you have to have a certain gene orientation, right? The neighboring gene has to be divergent. And of course, it has to be within a certain distance. Although we should say that we see this type of effect naturally affecting things that are over 2 kb away. So it's not like it has to be close, but it can't be, 10 kb away. Probably you're not going to get this effect. So yeah, I would say 7 to 10% of cases, people are probably misinterpreting mutant phenotypes or potentially misinterpreting mutant phenotypes because of these changes. But I do think, we can slightly predict where this might be a problem. And then if you fit those parameters, again, divergent orientation and close, then I think you don't really know when it's a problem and when it's not. So yeah, sorry, I don't have a more precise answer.
Perfect. Thank you so much. Yeah, I think with that, since we're on the half hour, I think we're gonna move on. But you do have the option to obviously answer the last question that just popped in chat. Okay. With that, thanks a lot, Gloria, for that really nice presentation. And yeah, we'll move on to the second presenter of the day. So basically, I'll give a quick introduction. So I'm really excited to introduce Rhiju for our second presentation of today's seminar. His lab has really been at the forefront of RNA structure prediction for many years, and as a core part of his research, Rhiju leads Eterna, an open science platform that crowdsources RNA design problems to thousands of players in an online game. Recently, this very same principle has been applied also in an effort to improve RNA vaccines. So a quick intro also to his background. So after his initial studies in particle physics and cosmology, he joined Dan Hirschlag's lab at the Stanford University for his PhD. Rhiju then did a very short postdoc working on protein folding predictions at the University of Washington with David Baker, after which he started his own lab in Stanford in 2009. There he continues to work towards better solutions for computational modeling and design of RNA molecules. Therefore, we will hear his latest update on the progress of RNA 3D modeling today. And without further ado, please join me in welcoming Rhiju for his presentation.
Thank you, Stefan, for the invitation to speak and for organizing. Can you hear me?
You're welcome. Yeah, we can hear you perfectly.
Yes,
fine.
Great. Yeah, thanks everyone also for joining in, all the RNA enthusiasts out there, and to the RNA Society for helping organize. For many years now, I've had a dream, which many of you might share. I'd love to be able to take any RNA sequence that I'm studying, whether it's a mRNA or pre-mRNA, or perhaps even an RNA therapeutic that I'm designing, and to be able to figure out which parts of the RNA might be forming interesting three-dimensional structures. And if so, what are those structures? And what do they tell me about the function? For example, this RNA, when it was finally crystallized, its structure showed us that it was a piece of RNA that is common in some slime molds, but it's analogous to selfish RNAs in other systems, and that appears to be what its function is. It's a parasitic RNA. My lab has primarily been working on this problem from the computational side, and over the years, there's been some progress. In blind competitions called the RNA puzzles, our lab, as well as others, have been able to make predictions, blind predictions of pretty complicated RNAs, like these riboswitches that sense vitamin B12 or adenylcobalamin. This is a riboswitch that senses SAM or S-adenosylmethionine. This is a riboswitch in the background, a T-box that senses another RNA, tRNA. And this is a riboswitch where we were challenged to predict the structure of the RNA, both with and without its glutamine ligand. And in each of these cases, what I'm showing you here are the blind models that we submitted in these RNA puzzles competitions. And we were elated in every case to find that a few weeks later, when the crystal structures were released by the experimentalists, that our modeling methods could recover or had recovered the global folds of these RNAs. Now, things still aren't perfect. In all of these cases, we had to submit 10 models, and this is the best of those 10 models, and the other models were not nearly as good. And if you zoom in carefully, you'll find that the atomic details are not correct in most of these predictions. But there appears to be progress. So why aren't you able to use this right now on your favorite RNAs of interest? Well, let me explain what's happening in terms of the recipe that we've developed along with others. And our methods share an overall framework that's really quite similar. So this is, in broad details, is pretty similar to what we do with our Rosetta and Farfar methods, but also what the Chen Lab does at University of Missouri, the RNA composer method from Martha Chachny's group, and the SimRNA methods from Gene Silico and the Buniecki's group in Warsaw. We will take a sequence that we're given, and we'll try to figure out its secondary structure, that is its pattern of Watson-Crick-Franklin base pairs. That defines the helices of the RNA. And then separately, we'll typically do a search to see if any of these sequences look like they might be matching previously solved structures. In this case, there's a T-loop from tRNA that appears to have recurred in this particular RNA target. And then we have computational methods that sometimes we call fragment assembly methods, where we mix and match the helices here and these other additional template motifs to create pools of often very large pools of models, hundreds of thousands of models. And then we will cluster these models and then pick 10 that we will submit to one of these competitions. And the thing that we don't always describe in detail is that most of these tests benefit from human inspection. So there's many methods for estimating the secondary structure of an RNA. And often we, as humans, as quote experts will often have to throw out the predictions that look weird, don't agree with other ones, or where we know that there's an artifact with one particular method. Similarly, in terms of scanning the sequences for motifs, we'll often do that by eye. Even here at the final step where we're trying to pick which models to submit, we'll often there's often human intuition, figuring out which of the models might be the best to submit. And starting about 5 or 6 years ago with the advent of deep learning approaches in other fields, we and others started to ask, well, could you bring to bear these more powerful machine learning methods to replace at least these human inspection steps? And we, in collaboration with Ron Dror's lab, first focused on this model selection step in this paper called Geometric Deep Learning of RNA Structure. We found that when we used a neural network called the Aries neural network to pick amongst our pool of models, we were able to make blind predictions, in this case shown here in blue, for a viral RNA that agreed remarkably well, almost indistinguishable from the crystal structure of this RNA that was released later. The best competing prediction actually was made by another method from our lab, but we picked out the wrong structure because we didn't apply Aries in this second set of predictions. Now, Aries still isn't perfect. Here's another case of an Aries prediction shown in blue against the crystal structure shown in green. And you can see they don't agree very well down here. There's a funny rotation that's happened. But here, it's the reason appears to be because the pool of models that we generated with these other upstream methods doesn't provide any more accurate kind of models. And so we started to think, well, could we now start to apply deep learning to automate every step of this structure modeling pipeline? And it wasn't just us. So after we published our paper on Aries in August of 2021, a slew of papers following up on our work, and especially inspired by the incredible accuracy of the AlphaFold structure prediction for proteins, started to come out over 2021, and especially in 2022. And if you read these papers, it just seems kind of amazing what kind of predictions they can make using essentially clones of AlphaFold, but for RNA. So the only thing that was missing at the end of 2022 was a rigorous evaluation of these methods. And the RNA puzzles competitions had gone under a slight hiatus and hadn't been available to test these. But fortunately, a new way to test these methods came about. Actually, it's an old way. Some of you might know that the AlphaFold method for protein structure prediction was evaluated in worldwide competition for protein structure prediction called CASP. And for the first time in 2022, we could evaluate whether we had an AlphaFold for RNA because we helped set up the first RNA category for CASP. And my group would usually be a predictor in this category, but because we actually organized it and provided some experimental structures, we chose to instead be assessors. And the results were surprising. Okay, so first of all, we were fortunate to have 12 RNA targets that were all quite biologically interesting. And we CASP elicited models from a field of actually over 40 predictors. So this is more than a quarter of the folks predicting for CASP, usually for protein structures, also decide to predict for RNA. And some of the models are really excellent. So I'm showing you here for 4 RNAs that were solved by crystallography, including the first RNA-only ribozyme that's been discovered in humans, the CPV3 ribozyme. There were blind models shown in blue that qualitatively agree quite well with the crystal structures that came out later, which are shown in green. The majority of the CASP targets came from cryo-EM, taking advantage of recent accelerations in that method for RNA-only structures. For 2 of these cases involved pieces of coronavirus genomes, which my lab and Rhiju's lab solved. And 4 of these cases were RNA nanostructures designed by Ebby Anderson and colleagues. And these are synthetic structures that have no homology to any existing natural RNAs, but it was wonderful to see, again, excellent models for all of these cases. The 2 difficult cases, I'd say, were RNA protein complexes. These are 2 bacterial RNA protein complexes solved by Zhao-Ming Su by cryo-EM. And here there actually were not good models that came in from CASP. So what were folks doing to get these kinds of good models? And was this just really done by 1 or 2 groups or by the entire field? So I'm not going to go into details of how we as assessors ranked the different groups, except to say that the ranking in CASP is done in a double-blind manner, where we as assessors actually don't only have code numbers for the groups who have made their predictions. And we look at a variety of metrics of accuracy and compile them together into these so-called Z-scores, and then rank groups by how high their Z-score was. It was pretty clear that there was one or two extremely good groups, and then 4 that really stood above the rest, which fill out this kind of plateau of intermediate z-scores below. We were curious to see whether these top 3 or 4 groups, whether they were each predicting well on all the targets, or maybe they were complementary in which targets they were predicting well in. And I'm showing you here a kind of heat map that's colored by whether each predictor did well in blue or poorly in red on each of the 12 targets. And actually, these top 2 groups did well, they were blue on essentially all of the 12 targets. Most notably, there are 4 targets in here that I mentioned already are non-natural. They're synthetic designed nanostructures. And particularly given the fact that in protein structure prediction, you typically need homologs from nature to solve a structure, it was really, really neat to see that these methods did quite well on these designed RNA nanostructures. But the surprise came when we got together for the conference. We got to read the abstracts and find out the identities of each of these groups. And I'm listing their names here. So out of the top 4 groups, 3 of them, the Chen group at the University of Missouri, RNA Polis, and Gene Silico, both based in Poland, were folks who I've interacted with for more than a decade. And they are the kind of classic RNA structure prediction groups. AI Chemi RNA2 was intriguing because it has the word AI in its title. But it turns out this group was led by Peng Xiang, who again has training in basic RNA. And based on their abstract, it seemed like they hadn't used any deep learning either. And so maybe that explains why they did so well on the design nanostructures, which usually deep learning methods require some kind of homology to previously solved structures. Well, here there were human beings in the loop who were helping create the structures. And to confirm all of this, we had a conference where we invited these predictors to speak. From Peng Xiang's talk, this is from the #1 group, he and his team applied both non-deep learning and deep learning methods, and they concluded that our deep learning method did not perform state-of-the-art structural modeling methods relying on scoring functions. This result is a bit different from what we saw on RNA puzzle tests, which were the tests that they used in their prior paper. Okay, so how are we going to get to an AlphaFold for RNA that all of us can use to go and look at RNA genomes or design therapeutics? Well, for deep learning methods or any kind of computational machine learning methods to work, you need 3 ingredients. One you need models that can be trained. And it was really cool to see that almost all the CAS predictors made their models available as open source code. So we actually now have about 18 to 20 interesting neural network models. You need data for training, and you need rigorous evaluation of those models. And fortunately, we have that now in the form of both CAS and ongoing RNA puzzles. So the thing that's really missing is the data for training. And this is something I think that many of you already know about. There aren't that many known experimentally solved structures of RNAs. There's, depending on how you count them, on the order of 1,000 compared to 100,000 protein structures in our structural databases. So how can we scale that up by the orders of magnitude that we need to get enough data to train computational models, say up to 100,000 or 1 million RNA structures? And here we're actually faced with the chicken and egg problem where we don't even have a list of 1 million diverse RNAs that we think might have interesting structures. So usually these structured RNAs are identified often by human beings who are looking at genomes by eye or with specialized algorithms and tools, and then they're putting them into databases like RFM as possibly interesting RNA structures. How do you scale that up by an order of magnitude or 2 to get a list of 1 million RNAs that you could then try to go after with methods like, for example, cryo-EM? Okay, so as one, in other areas of machine learning, the ways to get those big data sets has been often through crowdsourcing, by asking lots of human beings to create entries on Wikipedia, for example, or to classify millions or tens of millions of images. And we're fortunate in the RNA field to have a similar kind of project that brings together, that engages a community of numerous RNA scientists worldwide to look for interesting RNA structures. And that's a project called Eterna. If you haven't seen Eterna before, I invite you to go to eternagame.org. Here's the link down here. And let me just describe how it works. For 12 years, Eterna is a project whose tagline is played by humans, scored by nature. And we've had, every week, we have a challenge, a so-called lab challenge, where we invite people on the internet who don't have to have any kind of scientific background to send to us RNA structures or RNA sequences that they think will have structures or have functions that we're asking them to design on the platform. And then they can review each other's sequences, modify them, update them, and even vote on them. And we take the top ranked solutions, or when available, all the solutions, and we can synthesize them and characterize experimentally their structures and functions. And we return that data back to players for another round of design, or if they solve it, the community goes on to harder and harder problems. And through this virtuous cycle of gameplay and experimental feedback, this community has produced now over 30 scientific papers. Almost all of them have Eternal players as co-authors, and several of them are actually written by Eternal players. They're actually the first authors of the papers. And through these initiatives, we've started to collect, we've often collected really large data sets, which have been really powerful training sets for updating computation or developing new computational methods. I'd like to describe one case here over the next few minutes. In 2020, as many of you know, mRNA vaccines were developed in a record time for COVID. They were the first vaccines authorized. They turned out to be 95% effective against the first wave of COVID. And many of us who are here on the call have taken these vaccines, and it's helped us get back to our normal research and our normal lives. But one thing that I think many of you know is that the majority of people in the world were not able to get these mRNA vaccines. And that's due to a silly, almost prosaic problem, the poor stability of these mRNA vaccines, which is due to the RNA's susceptibility to degradation, prevents them from being shipped out in vials or prefilled syringes at room temperature or even under refrigeration. They have to be frozen at ultra cold temperatures to get to most parts of the world, or to get to where they need to be. And places like India or Brazil or Africa do not have those frozen cold chains. And so we, prompted by some pharma and biotech companies, asked the question, would it be possible to stabilize these RNAs against that intrinsic degradation process, which is caused by a chemical reaction that RNA does to itself called inline hydrolysis? And as part of these efforts, folks in my lab reasoned that we would love to have a predictive model for, given an RNA sequence and its potential structure, which parts of it are actually going to degrade. Okay, there's a heuristic that RNAs that are forming double-stranded regions, shown here in purple, are going to be protected against this hydrolysis reaction, whereas the parts in yellow or orange, which are looped out, can get into conformations where they can cut themselves. But what are the actual rules? Well, how do we get these patterns? And so we actually decided to crowdsource this to Eternal players and said, make us a whole bunch of interesting RNA sequences and structures that you think might be protected against degradation that we could use as building blocks for stabilized mRNA vaccines. And they presented, and they sent us a huge number of solutions. The main thing you're supposed to see from this diagram is that if you cluster the solutions by sequence, there are a huge number of clusters. There's an extraordinarily diverse set of submissions, and I'm showing here some example of structures here, which populated a large number of sequences that no one in my lab would have thought to include in a library that we would have designed ourselves. And then working together with Maria Barna's group, we developed a method to go and map out the degradation profiles for all these RNAs in a method called inline-seq, where we subject the RNAs to accelerated degradation conditions. We map out the fragments that result from these kinds of intrinsic cuts, and then using data processing pipelines similar to what we and others have developed for DMS or shape chemical mapping, we mapped out the profiles of degradation for these RNAs. And those were a very rich data set, enough for us to then take the predicted sequence, the known sequences and the predicted structures of each of these interesting RNA fragments, and to develop a very simple machine learning method based on a so-called Rhiju regression model that would allow us to then predict the degradation for arbitrary new RNA linkages. And again, I won't have time to describe everything we've done here, but using that model, then Eternal players were able to come up with ways to take an mRNA that would code for example, the SARS-CoV-2 spike protein, and redesign it to code for the same protein, have the same 5' and 3' UTRs, but where the coding sequence then folds up into a super tight structure that's predicted to have a low degradation rate. And then we tested experimentally and found out that indeed these RNAs have roughly double the lifetime compared to conventionally codon-optimized mRNAs for their chemical stability in solution, and they have similar and in some cases increased protein expression inside living cells. So this is described in a paper that came out last year. And what I love to flag here is, in addition to a bunch of Stanford authors, this includes over 100 authors contributed to this so-called Open Vaccine Challenge from Eterna. And then we also have 9 authors here who came from Pfizer. They tested these RNAs in their own hands for protein expression in New York. And so you have this kind of interesting, I think is the first paper that has involved both video game players on the one hand and citizen scientists joining up with, in this case, the biggest drug company in the world to check a scientific insight. Okay, so going back to this problem of how do you get large data sets, in this case, 1 million potentially diverse RNAs for training something like an AlphaFold for RNA, we're excited that to now be launching a new challenge on Eterna. We're calling it the Open Knot Challenge, where we're asking for, by the end of the summer, 1 million RNAs with interesting tertiary folds. We're to help focus some of the player community. We're providing algorithms that predict pseudoknots, and this is called the Open Knot Round 1 Challenge. This is happening right now. The first round is happening with 90,000 designs being collected by the end of May. And we have data for about 5,000 pilot designs that came in earlier this year. We are releasing those data later this week. So we invite any of you who are interested in this, or if you have friends who are just interested in science and maybe outside science, or you have nieces and nephews who are interested in taking part in science at a high school or college level, Eterna is I invite you to reach out to them and let them know about Eterna and this Open Knot Challenge. Every sequence that they put into this project will be synthesized and chemically mapped, and its structure will be checked experimentally. Okay, so some take-home messages here. In part 1, I showed you that I gave you a progress update on 3D RNA modeling. In the Cas15 competitions, we saw excellent models coming in from a few pretty great groups, but these predictions did require having humans in the loop along with their computational algorithms. In part 2, towards finding ways to get large data sets to potentially create fully automated 3D RNA modeling methods. I've told you about how we can crowdsource in a platform, in this case, Eterna, and how, as a proof of concept, that's led to predictive models for RNA degradation coming from thousands of human designs, and that resulted in a predictive modeling algorithm now in use for RNA therapeutics. And I just want to reiterate here, if you didn't see on my first slide, a disclosure, I'm a co-founder and I have shares in this company called Inceptive, which is helping take some of these insights into the market for helping stabilize RNA therapeutics. And then coming next, we have this Open Knot Challenge on Eterna, which we hope will feed into 3D RNA models that will be trained on 1 million novel human designs or discovered RNAs. So I'm going to thank the folks who did the RNA puzzles assessment with me, Rachel Kresh, Philip Pham, Rami Rangan, and all of these wonderful folks who we've collaborated with on the CASP assessment. And the MVPs of the Open Vaccine Eterna project are Hannah Weymouth-Steele and Anne Cladwang on the computational experimental side. And there, particularly for some of the more recent machine learning advances that have come from those data sets, I want to thank these folks, Do-Soon Kim, Andy Watkins, and others, especially folks in the Kaggle platform at Google. These are funding resources, and I'd also like to thank the Eterna players. These are the main developers of Eterna over the last few years. They're former players, and these are more of the folks who have joined us in recent years. So I'm going to, and then here's one who, an Eterna player who won the HBCU STEM Queen Award last year and was on the cover of Ebony magazine. And I thought, I just wanted to highlight that we have incredible people, many of whom are coming in through non-traditional paths into science through Eterna. That's something that I'm very, very excited about continuing.
Great. Thank you so much for that really exciting presentation and the updates on the recent RNA folding predictions. We have a couple of questions coming in the Q&A. So I'll start with Niels Walter, who's asking for long mRNA that needs to be translated. The challenge is, of course, that a single cut is typically sufficient to lead to loss of its utility. Would the stability at room temperature during transport be linearly dependent on the number of vulnerable nucleotides? Yeah,
so I think that linear relationship longer RNA has more real estate for having even one cut. So we qualitatively or semi-quantitatively check that linear relationship in that paper. That totally holds. And so yes, so the problem of RNA stability is actually not such a big deal for silencing RNA therapeutics. And then it gets a little bit worse for therapeutics that are trying to package like guide RNAs for CRISPR. But it gets really, really bad once you have RNAs encoding for the spike protein. So those are 4,000 nucleotide RNAs or RNAs that encode for like CRISPR, the CRISPR-Cas9. So I think this is going to be a problem that we need to solve as a field if we want to have equitable RNA medicines.
Yeah. So we have another question from Marcin Magnus. So the question there is, Rhiju, are you trying to get 3D structures with cryo-EM, I guess, for the 1M designs? I guess for some, yes. For how many?
Yeah, so we're currently the great thing about cryo-EM is we're now in a mode where we can solve something like 10 or 20 structures in a single study with cryo-EM. Most of that time is spent on screening conditions for grid preparation, as some of you know. One thing I'd really love to do, and I should say, if anyone out there is interested in postdocs or PhDs, I'd love to find a way to do massively parallel cryo-EM. In a given cryo-EM session now, we can collect data on like 100 million particles, but you only need 100,000 particles to solve an RNA structure by cryo-EM. So if you could somehow put in 100 or 1,000 RNAs at once onto the grids and barcode them, structurally barcode them, then you should be able to solve a lot more structures at a given time. So that technology is not ready yet, at least not in our lab or in Waz lab, but maybe in the next 1 or 2 years, and then that would let us look at all the designs coming in. Maybe, I don't know about 1 million, but we'll see. Thousands, maybe.
Okay. We have some more questions there. One is, will if they're not considered non-coding RNAs?
Let's see. I don't understand that question.
I wasn't entirely sure. I thought.
Oh well, Eterna considered non-coding RNAs.
Yeah. Sorry.
Yeah, totally. We're delighted with non-coding RNAs. Yeah. I'm having a debate right now with some folks, some of the top Eterna players, where I'm like, I think introns in metazoans are full of super interesting RNA structures, and I have my pet reasons for why I think that's the case. And of course, most of the genome is non-coding RNAs, and a lot of that's introns. But then there's some players who are like, no, they think the coding regions have the most structure for really distinct reasons. And this is why I love this community, because it's just like very different viewpoints, and we're going to test all the viewpoints. So yes, if you want to come in and run non-coding RNAs, I'd be happy to be on your team. But if you also want to go and just find windows of coding RNAs that you think might have structure, that's totally great. Yeah.
Nice. The next question is if there are any plans to integrate modified nucleotides in the RNA designs.
Yeah, actually, so the open vaccine stuff included pseudouridine and 1-methyl-pseudouridine. And so there's a little bit of that is discussed in the paper. And it seems like computational methods are such that if you get enough data on RNAs with modifications, then you can infer the appropriate folding parameters for those modifications. We're not doing it right now, but I think in the next year or two, that will be including modifications in the folding of Eterna and in the prediction of 3D structure, I believe should be doable.
Cool. I think we have time for 1 or 2 more questions even. So one more is Moderna shows that integration of stem loop double-stranded RNA in coding sequences of spike protein mRNA vaccine. Do you think that enriched double-stranded RNAs may reduce mRNA translation elongation as a cost of stabilizing mRNA?
Yeah, it was a surprise to see that including we call these super folder mRNAs. So the ones that came out of Eterna are actually way more double-stranded than the Moderna designs. So we assumed that they would really suck as potentially like for protein expression. And they probably do reduce mRNA translation elongation. And we see some evidence for that from ribosome profiling. Or just polysome profiling. But some of them produce more protein, apparently because they last longer inside. We designed them to last longer outside cells to be chemically more stable, but they also, for reasons that we don't fully understand, appear to be, some of them are more stable biologically in cells. Maybe they're protecting against recognition by the factors that lead to, the various factors that lead to mRNA decay. So that was a surprise. And I don't really deeply understand how that works yet.
That's interesting. I think we do have time for that very last question, since I think that could be answered quite quickly. So it's really just asking how long RNAs are that you will test by cryo-EM.
Yeah, the current Eterna lab puzzles are 100 bars. That's kind of in the sweet spot, a reasonable sweet spot for cryo-EM. They're actually very small for cryo-EM, but it turns out RNA has good contrast in cryo-EM for reasons we don't understand. And so even though those are just 30 kilodaltons, you can see them.
Cool. All right. I think we're on the hour. Thank you very much, both speakers again, and all the interesting questions that we got in with this seminar. And yeah, looking forward to the next seminar in 2 weeks.
Thanks, everyone.
Thank you.
Thank you.